So I'm very happy today to talk about some of our work where we analyze genomes and where we identify some signatures of interspecific hybridization. And I would like to start by introducing the model organism that we work with. So Dave already mentioned that we work with a fungus called Septoria tritici, previously known as Microviruses graminicula. It's a pathogen, foliar pathogen of wheat, and it's characterized, it causes these necrotic lesions. The small black dots here are the asexual fruiting bodies of this fungus here. And I thought that it's relevant today also to start by saying that we are living through a pandemic, Corona. We have in agriculture, we are experiencing also pandemics. We have experienced pandemics the last centuries, and many of us, we are working on some of these diseases that cause, these pathogens that cause disease on pathogens, on plants. So I'm showing you here a world map that shows the average production of wheat worldwide. And as you can see, on almost all continents, we are cultivating wheat. And Septoria tritici is one of these, let's say, more pandemic pathogens that can be found worldwide where wheat is cultivated. So some of the questions that I have been interested in since I was a PhD student actually is the origin of Septoria tritici. So this, you can say, pandemic pathogen, where did it come from? And we have over the years been able to, let's say, geographically localize the center of origin of this pathogen here, but we still have a lot to do in terms of understanding the processes whereby this pathogen here emerged, the speciation of the pathogen. But surely the speciation of Septoria tritici is tightly linked to the adaptation to wheat as a host. So one of our goals is also to understand the interaction between Septoria tritici and the wheat host, and in particular those traits that determines host specificity of this pathogen here. And one issue that is in particular here in Europe, where we have a very intensive cultivation of wheat, and we apply a lot of fungicides to control fungal diseases, including Septoria tritici, a big problem for us is the evolution of fungicide resistance and a very rapid spread of resistance mutations. So something that a big concern is how in the coming years we're going to be able with fungicides, with chemicals, to control Septoria tritici. So understanding mechanisms of rapid evolution in this pathogen is very important in terms of designing future control strategies. And studies of evolutionary mechanisms starts with population genetics. And so a lot of the work that we are doing and work that has been done in Septoria tritici has focused on the population genetics of this fungus. And I think it's not an underestimate to say that this has been a primary model in population genetic studies of fungi. And I have listed here work of Bruce McDonald, who has been, let's say, pushing research as Microviruses graminicula, Septoria tritici in the past 20, 30 years. So one of the big, let's say, achievements of Bruce has been the collections of isolates from all over the world. And he has over the years been genotyping these isolates with different marker systems, and with that been able to record genetic variation of this pathogen at different spatial scales, from single lesions on a leaf to variation within a field, within a region, continent, and on a global scale. And with different population genetic methods, with evolutionary predictions, we've been able to reconstruct the historical gene flow of Septoria tritici. And so what we have learned over the years here with this global collection of Septoria tritici is that we find the oldest populations that have the, let's say, that the highest amount of genetic variation, we find those in the Middle East. We have been then reconstructing gene flow and historical gene flow of this pathogen here and find evidence for a very early spread of the dissemination of this pathogen from the center of origin into Europe and to Asia. And then only very recently, in the past few centuries, to what we can call let's say New World continents, and across continents to South Africa. And when you see this map here, you will see that this mirrors the map also of the dispersal or the spread of wheat cultivation on a global scale, first from the Middle East, from the Neolithic, from the Fertile Crescent to Europe and Asia, and then only much later with European colonization and with the spread of wheat cultivation to the Americas, Australia, and also here to South Africa, where many of you are sitting. So in terms of understanding the origin of Septoria tridici, we have been focusing much of our research in the Middle East. And this is a particularly interesting region for all of us who work on cereal pathogens, because it's also the geographical center of origin of many of our important crops, of our cereals, of wheat, barley, oat, and rye, which are crops that were domesticated during the Neolithic revolution 10 to 12,000 years ago. So what we have been doing is that we have been hypothesizing that Septoria tridici has originated from a host jump from wild plants in this region of the world. And so we have been collecting Septoria isolates from wild grasses. And I want to mention here very important collaboration partners, Mohamed Yavanika and Ali Reza Alizadeh, who are both plant pathologists working in Iran. Ali Reza was a student of Mohamed and now has his own associate professor position in Tabriz, and he goes every spring to the north of Iran, to these beautiful mountainous regions here, and he collects wild grasses for us that have symptoms of Septoria disease. And we are now focusing, or Ali Reza is focusing now his collections on wild relatives of wheat and barley. So you will see here, this is Aegilops tauchi, which is an ancestral species of wheat, and that also bears symptoms of Septoria tridici. And I will not talk about that today, but we have actually on these wild wheat species, we have found a subspecies of Septoria tridici that is very specific to these wild wheat species here. But so we use this diversity of Septoria species to study the evolutionary history of the pathogens and their population genetics. And we have over the years described new species of Septoria, Adabilia, Septoria brevis, and Septoria pseudotridici that infect different wild grasses. And this is an awful phylogenetic tree because the branch lengths are so short here, so it looks very messy. But that's actually the beauty of this system here, is that these are very closely related species that infect different hosts in different environments. Septoria tridici in an agricultural environment, and the other species here in natural grassland vegetations. So that allows us to study the speciation history of these pathogens, and also their population genetic that is associated with being a pathogen on wild plants and being a pathogen on a domesticated or cultivated plant. So we have been using this data also to date the speciation event of Septoria tridici, and here we have been using evolutionary analysis, coalescence analysis, where you identify the time of the most recent common ancestor of the different species. And what we have found evidence for with independent methods and different types of data sets is that the speciation of Septoria tridici has been very recent and very likely coincide with the domestication of wheat. So we think that there is strong evidence for this pathogen here actually having speciated along with the domestication of wheat, and subsequently it's taken a ride on the dispersal of the host on a geographical scale. So the way that we study So I mentioned that Bruce McDonald has been doing population genetics for many years with these pathogens here, from using RFLP markers, microsatellites, DNA sequence loci. And today, a lot of the population genetic analysis we base that on SNP data from whole genome sequencing. And so I've listed here in parentheses the number of isolates that we have, from which we have obtained genome sequences. And this provides us then with a, let's say, a fantastic resource to study genetic variation within and between species. So I have a small, I think it was Dave who mentioned Liesel before. So maybe some of you know or remember Liesel, who was a student at FABI and now is a PhD student with me. She's actually finishing her thesis these days. Liesel has a small paper here where she has described the bioinformatic pipeline that we have set up to analyze genomes of the Septoria species here. We have namely a challenge, and the challenge is that these fungi have a lot of structural variation. So they have extra chromosomes that we call accessory chromosomes. And in addition to that, they have in their core chromosomes, they have accessory regions that show presence-absence variation between isolates. So that challenge, let's say the characterization of genetic variation only based on reference-based mapping approaches. So what we do is that we use Illumina sequencing to call SNPs, but we also generate de novo assemblies based on both Illumina sequencing and also PacBio sequencing. And we align these genomes in order to characterize also the structural component of variation between isolates and between species. I just wanted to mention that. But otherwise, Septoria, these species are typically, let's say, have a typical dotidiomycete genomes around 40 megabase, around 10,000 genes. And then they have this large number of accessory chromosomes that are particular, I would say, what we know so far for this group of dotidiomycetes. Our information on the genetic variation, we use that to study the mechanisms of evolution in these different Septoria species. And we are particularly interested in characterizing the mutation rates along genomes. We study recombination and recombination rate variation. We and others have been looking at local and global patterns of migration in time also. And then we also make inference about the type of, or the evolutionary forces that act on this genetic variation. For example, natural selection and neutral processes such as genetic drift. And we also use this data here to make inference about the demography of the pathogen. In particular, we're interested in past changes in the effective population size of these different pathogens. So what this small map here shows are processes that we think about as shaping genetic variation within populations. But something that popped up and actually sort of seems to play an important role in this group of fungi is actually interspecific recombination. So normally here we consider recombination as a process that occurs within the population, but we actually happen to find signatures of this interspecific recombination, hybridization, and also that this plays an important role in shaping genetic variation in these genomes. I would like to take you back to my postdoc time here and the first genomes that we sequenced of Septoria tridice and pseudotridice with Illumina sequencing. So this is this data dates back to 2008, 2009, 2010. It's not that long ago, but still, I'm sure that today I cannot impress many of you with these. We sequenced at that time 3 genomes of Septoria tridice and 5 genomes of Septoria pseudotridice, and we call that population genomics. Back then, 10 years ago, that was a lot of data. So what I did during my postdoc was that I would analyze genetic variation along genomes, and I would plot it like this. This is an alignment of chromosome 4, and it shows 3 different values here. First, dxy as a measure of the divergence across a chromosome, the divergence between these 2 species. And then it shows pi, the nucleotide variation for each of the 2 species, for pseudo-3DC and for 3DC. And one of the first observation that we stumbled across was these long stretches here where there is no variation in the genome of Septoria pseudo-3DC. And in fact, we would find on some chromosomes stretches up to 100 kb, where among these 5 genome sequences, there was no variation, not a single nucleotide polymorphism. And we would, of course, use PCR and Sanger sequencing to validate this pattern. The interesting thing was not only these long stretches of non-variable sequences that we could confirm, but was also the pattern of variation that we would find interspersed between these non-variable regions. So to characterize this pattern in pseudo-3DC with our, at that time, 5 genome sequences in better details, we chopped up the genome in windows of 1 kb, and then for every window, we would characterize if the window was a variable or non-variable window. And for the variable windows, we had a very interesting observation, and that was that the way that the variation was distributed was always as 2 haplotypes. So there would always be either a pattern where one sequence would be different from the 4 other, and these 4 would then be identical to each other. Or there would be, the alternative here would be that there would be 3 sequences that would be identical to each other and different from 2 sequences that would be identical to each other. So we always had these 2 haplotypes in the variable windows. So if you have 5 sequences and you put them together in this way here, you end up with a total of 15 possible topologies. And so what we did was we characterized along the genome alignment these windows as either being non-variable or belonging to one or the other of these these 15 topologies. So we would have a map that looked like this. So the only way that we can, and again here I recall that we have here very, very long stretches with absolutely no variation. So where can such a pattern come from? The only scenario that we could come up with is that Septoria pseudo-3DC has originated from a hybridization event occurring between only 2 fungal individuals, which at some time in the past have crossed with each other and generated a hybrid swarm, hybrid population. And this hybrid population has then been crossing among itself, but has never back-crossed to any of the parental individuals or to anything else. That's the only way, the only scenario whereby we can explain these long regions with absolutely no variation. And of course, then the variable segments, these 2 haplotypes that we identify, represent those sites in the genome where the fungal individuals have inherited either the sequence from parent 1 or from parent 2. So we could go a bit further in terms of understanding when this hybridization event had occurred, because we could look at now at the divergence between these 2 haplotypes, the parental haplotypes, and that's a 3% divergence between the parental haplotypes in comparison to a 6% divergence between Septoria 3DC and Septoria pseudo-3DC. So the scenario that we think is that there was in the past, and this is what we know has been around 10,000 years ago, there's been a speciation event. This guy here has then specialized and spread with wheat. This guy has been infecting wild grasses in Iran, and at some point has diverged into 2 reproductively independent lineages, which have then very recently come together, forming this hybrid Septoria pseudo-3DC. We wanted to date the event of this hybridization event. And the nice thing is that this mosaic pattern that we have here. So this, of course, reflects the recombination events that have chopped up the genome and scrambled around the genetic variation. So having information about the average length of these small fragments here, where we have consistently one topology, we can use that information together with information about the recombination rate that we had estimated in another study. We know this is more than 40 centimorgan per megabase. And so then we could do having also information about the genome size. We know how many, let's say, crossover events there will be per sexual generation in this species here. And then we can do a quick and dirty calculation here to compute how many sexual generations would it take to chop up the genome in the pieces that we observe. And so we find something that is very recent, so very likely less than 400 sexual generations ago. So Septoria pseudotridici, so this data was, as I mentioned, only based on 5 genomes. Since then, we have sequenced more genomes. Actually, we have now a data set of 27 genomes in total of Septoria tridici. And this is actually a data set that has been lying around for several years, and there's none of my people who are really interested in picking up on this data, and that's because we actually cannot really add any new information, or very little new information, having 27 genomes compared to having 5 genomes, because we find consistently this pattern here where we find these non-variable regions, 27 sequences that are identical to each other in very long stretches, and that are interspersed between regions where we have 2 clearly defined haplotypes. The new thing is now that we start also to, with this new data, we can also identify new mutations that derived mutations that are accumulating now since the hybridization event. So I always, since then, have been considering Septoria pseudotridici as an outlier. And I'm sure you know each of you have your own favorite model organism that you work with and that you think you know by heart. And for me, it was always Septoria tridici is a special, it's a hybrid, but otherwise hybridization doesn't occur in this group of fungi, and certainly not in Septoria tridici that we know so well. There had never been any mentioning or thought about hybridization also occurring in Septoria tridici. So this, let's say, impression of mine changed, and it changed together with a very talented postdoc who worked with me, Alice Ferté. It's now actually also collaborating with Brenda, so I think that that's going to be some very interesting thing also coming out of that now. Alice is now in Zurich for a second postdoc. But when she joined my lab, she started to work on our population genomic data set of Septoria tridici, and she was she's a very thorough scientist, and was interested in understanding really when you filter your data in different ways, what is it that you throw out, what is it that you keep, and one thing that she stumbled over when she had done a SNP filtering that she thought was satisfying was that she would find these strange peaks here. And again, here we have chopped up our genome alignment in windows of 1 kb, and then we have counted the number of variable positions, and you can see here that we actually have some peaks here where more than or half of the sites in a window are variable. And we had not seen these regions here before, because usually we would just kick out these with our filtering and consider that as poorly aligned regions. And Alice wanted to understand, why is it that I do a very good SNP filtering and I keep having these weird regions? What are they? And I will take you through an alignment here. So what Alice did was that she did a lot of manual inspection of genome alignment. That's something that we don't usually do in genomics, but sometimes that can actually be very informative to go back and look at your alignment, and of course not the full genome alignment, but on some selected loci. And so when Alice would look over the genome alignment, it would look in a normal window would look nicely like this, and you will have your polymorphisms that are segregating as expected. And then when you start to move into some of these more variable regions here, we identify a very interesting pattern here. And what we can see is that, or at least what I recognized, was something that looked very similar to when I had been looking at the genome of Septoria pseudotriticale, namely that we had this pattern of two distinct haplotypes, and with very similar sequences within these haplotypes, but the two haplotypes being highly divergent from one another. So I'll just move my small window here. And so on the other side of these highly variable regions, then you sort of fall out again in a normal pattern of genetic variation. And so we of course confirmed with Sanger sequencing across these regions here that they are really there in the genome. So we, building on our previous experience with pseudotriticale, we hypothesized that these highly variable regions in Septoria triticale also could reflect past hybridization events. And to address that, Alice took a phylogenetic approach, and again here using this approach of walking along the genome alignment and generating phylogenetic trees. So here we used PacBio sequences, and we aligned 3 genomes of Septoria triticale with genomes of pseudotriticale, brevis, adabilia, and Septoria passerini. And this small phylogenetic tree here in one of the normal regions, this is how the species tree look. This is what we expect. And this is what we find mostly as we walk along the genome alignment. And then we will again stumble over these highly variable regions. And we will see there that our gene tree, or the phylogenetic tree that we can assess in these windows is incongruent with the species tree. So in this case here, one of the Septoria triticale sequences cluster very closely here with the adabilia sequence. And so we find here a pattern that's, let's say, where we find a high, let's say, a pattern of these highly variable regions showing incongruence in the phylogenetic relationship of the Septoria species and supporting our hypothesis of hybridization. And so what Alice additionally did is that she would sort of quantify in how many of my genome windows do I get the normal expected phylogenetic tree. And there's a huge excess of this normal phylogenetic, the species tree consistent phylogenetic tree in normal windows, whereas in the highly variable regions, we have an excess of these alternative phylogenies that do not follow the species tree. So what we interestingly also find here, so you can imagine that then of course the big question is, so does this have a, let's say, an impact on some sort of functional relevance to have these highly variable regions? And we are not there yet where we can really demonstrate the functional relevance, but at least we know that these highly variable regions co-localize with more than 1,000 genes, and a small number of these genes are actually effector candidates. So here you can imagine that you have a scenario where you have 2 different Septoria triticale isolates that have these highly different composition of effectors, which we hypothesize have a functional relevance. So just to summarize here, and I really want to encourage all the young participants who are working, who are those of you who are sitting at the computer and analyzing genome data, to always, when you have some weird thing, when you filter out data, always to have an extra look at these things, because we were not expecting to find traces of signatures of hybridization in the Septoria genome, and it was only because of Alice's very careful manual inspection of the genome alignment that we could see what these highly variable regions actually are. And with our phylogenetic analysis, I think this provides us with a very strong evidence for frequent hybridization occurring between Septoria tritici, or that it has frequently occurred between Septoria tritici and the other wildgrass pathogens. And for us, of course, an important question is the adaptive role of introgression. Could there even be selection for once in a while outcrossing with fungus on another host? And I just want to say lastly here that we do occasionally in some of the genes that we study that come up from other studies, we do occasionally actually see the signature of this introgression. And Klaas Jotanos worked with me some years ago on the role of RNA interference, and he looked at different genes encoding Argonaut proteins. And Klaas would make phylogenetic analysis of these Argonaut genes. And what you see here is also the sequence of Septoria tritici here, clustering in two highly distinct groups here, in this case here with Septoria pseudotritici. And you see also here Septoria tritici clustering here with Septoria adabila. So maybe this reflects a region that is very frequently involved in or experiencing this type of introgression. And I also there want to say that when Klaas, when he was working on this, we were often discussing what could we be doing wrong with our phylogenetic analysis or our alignment, because this just cannot be true. But now we are seeing for more and more genes that we are analyzing in Septoria tritici, we see this pattern of very highly differentiated alleles. And lastly, I also want to mention, and I'm very happy that Dave, you made advertisement for our papers on bioarchive before when he introduced me. And one of the papers that is on bioarchive is a paper that describes the diversity of transposable elements in Septoria tritici and between closely related species. I will not go into details about this study, but just want to mention here that one thing that has been impressing me with this data set is a very, very dynamic composition of transposable elements and very little species specificity. So it looks like these different Septoria species, although they have different amounts of TEs, they share very much the same families. And what we think is that hybridization is one mechanism where they can, where transposable elements can move between species. So, so with that, I'm actually at the end here, and I would just like to thank you all for your attention, and also thank you to my group. Let me see, Alice, she's here, and Cecile, who's been doing the TE work here. And I'm very happy to take any questions.
Okay, you can't see myself. Okay, thank you very much, Eva. That was a fascinating talk, and apologies to everyone about the hacker which came in. So that illustrates that there's a zoo out there, and there are 2 points from your talk. On the surface of a wheat leaf, it's obviously a zoo out there as well, where there's all sorts of intimate interactions going on between the different isolates there. And the second thing which you've illustrated in your talk is that if you ask the right questions of genome sequence information, you can really get some very interesting insights. I would like to start off with one question, and then before we get on to other people's questions. You talk about in this latest part in Septoria tritici that there are these variable regions. Are they associated at all with those accessory chromosomes, or are they spread throughout the genome?
So that's a very good question, Dave. So we have actually. So the thing is that the accessory chromosomes are already, let's say, a challenge to work with, and often they are very repeat rich. And so we actually, let's say already we have many more inconsistencies in our genome alignment, which makes it very difficult to really, let's say, distinguish where you have the normal to the highly variable regions, because the alignments per se is much more fragmented. But what I looked at when I worked with septoria pseudotridice, so there was, has there also been recombination between accessory chromosomes of the two parents, or could it be that they have inherited a chromosome only from one parent? And indeed, we do find accessory chromosomes in septoria pseudotridice with almost no variation for an entire chromosome. So that certainly suggests that that this process also affects the accessory chromosomes. I should say that in general, the accessory chromosomes have much less recombination, and that's because there's much less, let's say, I think that it's more difficult to have homologous recombination because the sequences are evolving rapidly and are less similar to each other. So.
Thank you. Let's open the floor. Can people ask questions, or is there any questions in the chat box?
So we have allowed the participant to unmute yourself and turn on the camera. Would you like to ask the question directly? But while we're waiting for that, Eva, thank you very much for the interesting talk. So the evidence you suggested that hybridization could be the cause that lead to the sharing of transposable elements and introgression related to transposable element between species. Sometimes we will see that, like, in a group of fungi, there's only one species that got a very dramatic extension of transposable elements. So I wonder if there are other type mechanisms that also help the spread of these transposable elements. But do you think otherwise from hybridization between species that having it, do you think we have other dominant way of transferring this TE across very distantly related fungal species?
So you're asking about the mechanism, really, if hybridization can be a mechanism where these fungi can transfer transposable elements between species?
Yeah, so hybridization could be the case here. I think that's quite evident. But do you think that maybe there's also other mechanism as well where there isn't hybridization involved? What would that mechanism be? Do you think of other type transferring TE between distantly related fungi?
So you're asking if what other mechanisms could transfer?
Yeah.
Yeah. I mean, this is something that we, indeed, because we have this very dynamic landscape of transposable elements, that we have been thinking a lot about how do they acquire these elements, how do they get into the genome. And I mean, there is a lot of literature out there on let's say, on the dynamic of TEs. And another possibility is are viruses that can bring in DNA transposable elements and can move that between species. And I think that it, of course, also relates to the discussion about how much horizontal gene transfer is there and what is the role of transposable elements also in a horizontal gene transfer. But I think that we know very little about the role of microviruses also in moving around such repetitive elements. Yeah, so I'm reviewing currently a proposal about bacteria that are associated with fungal hyphae, and I think that this is also this thing on the interaction of fungal hyphae with other microorganisms, not only viruses but also bacteria, and the possible exchange also of DNA is something that we know very little about, but could be a mechanism. But for sure here, I mean, what surprised me was this lack of really much signature of species-specific invasions, but much more that you have isolate-specific invasions. And so I think there is a lot of dynamic, and that's, of course, also why these fungi rely on having genetic tools, mechanisms to control transposable elements, such as, for example, repeat-induced point mutation, this RIP mechanism that is in many ascomycete fungi.
Thank you.
Go on. Answer the question. So there's a question from Pedro Kraus. We had the Pyranophora parastagnospora example. Any evidence for gene flow from Zymo-septoria to other fungal genera on cereals?
Yeah, so so actually one thing that so first of all, hi Pedro, it's nice that you are also there. So one thing now I showed you these nice example of the phylogenetic trees here, and you will see here it always sort of, what I'm showing is always that there is a clustering together with another Zymo-septoria species. But for a very small proportion of the windows, we actually find that one Zymo-septoria triticale sequence, it sort of falls way out of this, let's say, this genus here, and where we have not really been able to assert where it fits. And there still lies a project there to investigate that. But certainly, I think that this is also something that can happen, that the distantly related species can hybridize, and perhaps the immediate hybrid is very unfit in its environment, but maybe it serves as a transient bridge for genetic transfer between species.
Thank you. Pedro, I hope that answered your question. Can people put up their hands to ask questions, or?
Yes, they can unmute themselves.
Are there any? Okay, so I think if you want to ask questions, please unmute yourselves. Okay, here's question from Brenda. Have you evidence of mitoviruses in Zymo-septoria triticale?
Yeah, that's a wonderful question, Brenda. And I mean, we have never looked on microviruses. I mean, it's in the back of my head that one day we should try to do that. I don't have much experience there with microviruses. I know there was once a Brazilian colleague who was very interested in Zymo-septoria genomes, and he was studying microviruses and said that there were some very particular signatures of viruses in this group of in the genomes of this group of fungi. But yeah, I'm sorry I cannot answer it better, because I think it's such an important question. I think it's a field that is expanding now on understanding more the role of microviruses. And I just can mention, I heard recently a discussion, some colleagues that work on this Pseudogymnoascus destructans, which is a fungus that causes white nose syndrome in bats in North America. And you may have heard about this bat fungus. And there they are now. I think there is some new studies that sort of investigate the role of a microvirus on virulence on this clade that has invaded in North America and is so much more aggressive than European Pseudogymnoascus destructans isolates. So possibly viruses can also play a role in virulence and pathogenicity of fungi.
Thank you, Eva. So Brenda, I can add to that in actual fact, in Tanya's MSc, we found some microviruses in the Cercospora infected maize, and we published that in European Journal of Plant Pathology just a couple of months ago.
I did see that, Dave. That was lovely stuff.
And that was another wonderful example of serendipity.
Yes, no, that was I read that article with great interest. That was lovely. Yeah, I mean, I suppose there was a point when there were a very small number of us actually looking at microviruses, but I think microviruses might be about to come into their own. There's an example actually of using a double-stranded RNA microvirus actually to potentially suppress pathogenicity. So yeah, I think there's some more stuff out there that we should be looking at. Thanks, Eva.
Any other questions from anyone?
Dave, I have one.
Yeah. Bernard's got a question. Do you want to go ahead from there?
Yeah, go ahead from here.
Okay.
Thank you from my side also for a really fascinating talk, much enjoyed. So this initiation event that you've timed to the domestication of wheat, so 11,000 years ago, it's really fascinating to see that. But surely these processes are still continuing. I mean, the selection pressure must be many times higher today for such evolutionary events in many other pathogens. Do we see evidence of that, of similar emerging pathogens where similar, more recent processes has happened?
So we have recently started a new project where we look at Cercospora peticula, that is a pathogen of sugar beet. And the interesting aspect of sugar beet is that it was domesticated very recently, so just a few centuries ago. And we find Cercospora peticula also on wild beet. Beta vulgaris maritima, I think it's called, that we can collect along the Atlantic coast here. So we actually have a study there where we want to see if we can catch the same process of host specialization as a consequence of recent domestication. So as I mentioned also. Oh no, actually I didn't mention that, but then there is Septoria passerini that we always use as an outgroup. And Septoria passerini is a very, not very studied pathogen because it doesn't occur very frequently. It's a pathogen of barley, but there are some years where it pops up. And so there we are now starting to collect Septoria passerini from wild barley. And of course, being interested in see if there's a parallel, let's say a parallel scenario where the speciation on barley has coincided with the domestication of barley.
Fascinating. Thank you, Eva.
Thanks. There's just a comment from Jeff Dean. Thanks for a fascinating seminar. It was a great way to start the Thanksgiving holiday here in the US. So that's impressive that some US people are not already on their Thanksgiving holiday. Pedro has a question. There are at least two groups. New Zealand is publishing new species in the Cercospora beticola complex. You also need to look at Cercospora api on celery. That's a suggestion from Pedro. Okay, you got that? Any other questions from anyone? Eva, I want to just ask about your, where you actually have those huge regions which are not variable. And so is there some mechanism in the fungi to keep the sequence very, very conserved? Or is it just that there hasn't been enough time for you to see any differences in those regions? Is there something funny going on that it's so heavily conserved?
Yeah, so I think indeed that it's rather, I think that actually conserved is not the right term, but it's more that it's a very recent event. So there has not been time enough for new mutations to accumulate. And so in a way, I think that Septoria pseudotriticale is a very unique example of a very, very strong bottleneck that has been associated with this hybridization event. And for the pathogen must be a very, let's say that, you know, that's not very, very good for a pathogen to have such long stretches where you have no genetic variation. And so relying there only on the de novo accumulation of mutations. And this is what with this larger data set where we have 27 genomes, where we now start to identify single nucleotide polymorphisms popping up. So one thing is also we had one collection one year where we collected this material in Iran, and we actually have not been able to find Septoria pseudotriticale again, which maybe also suggests that it's a, let's say, a very very limited in its dispersal and host compatibility.
Great, thank you. Okay, so any last questions from anyone who would like to unmute themselves? Or Eva, I'm tempted with one last question about host specificity. So what is your take in Zymo septoria, which seems to go into the stomata, like the Cercospora? I think that's correct. So do you think the host specificity on these different grasses is really related to just the suite of effectors inside that are expressed inside the plant, or do you think there's some possibility that host specificity can be due to some surface recognition? Do you have any feelings on that?
Yeah, so there are a number of resistance genes that confer a qualitative resistance in Zymo septoria tridici, and there was a paper last year, I think, the STB6 septoria blotch 6 resistance gene number 6 that was cloned and characterized, and this recognized and let's say a protein called AVR-STB6 that is produced by some Zymo septoria tridici isolates. So there are certainly also examples of, let's say, virulence factors that are recognized in this system here. Yeah. But otherwise, what we see is really that in the incompatible interaction of Zymo septoria with let's say with a non-compatible host, is that the infection is really blocked in the stomata. So there's something very early that recognizes the fungus and prevent further colonization. So we have actually never had, with several combinations of plant genotypes and plant grass species, weed species, different isolates of Zymo septoria, we have never had the scenario where we have the biotrophic colonization of the fungus and then just not the switch to necrotrophy. It really never gets further than the stomata if it's incompatible.
That's very interesting.
Mm-hmm.
Great. Okay. So thank you very much, Eva. That was absolutely fascinating. And I think now is the opportunity for Bernard just to finish off and say some words of thanks. Thank you.
All right.
John, do you want to move the camera across?
That's right. I'll put my camera. I'll put my computer camera on. I've just asked Tuan if you could share a screen for us there, a screenshot of our annual card. Perhaps capture some of what I'd like to say. But let me start again by also from my side thanking you again, Eva, for a really fascinating talk. It's a wonderful way to start Thanksgiving, but for moving into this festive season and to end this set of seminars for us this year. So thanks to Tuan. So Tuan's just share there every year at the end of the year we make a card for this festive season to just send good wishes to our friends around the world and best wishes for the new year that we're heading into. And I'd like to use this opportunity to also do the same. If this year has taught us anything, then it is how much we need each other and how much we've missed each other. And Eva, as you said, it would have been lovely to have you here, and it wasn't possible now, but we hope it's possible soon. And we hope that what we've experienced this year will be in the near future, something that we can put behind us and look ahead to a point where we can interact in person again. But as Wynand has said, we've also, of course, had this kind of interaction has been so fantastic. And I think for our students, for example, I think they've had as much or even more opportunity to interact with colleagues and leading thinkers from around the world than they've had in previous years. So likely, even if we can see each other, we'd like to continue to keep doing what we've done. But I wish all of you, I thank you for joining us for this seminar. I thank you for joining us for many through the year, and for everybody who's taken hands to keep the connections through this year. We look forward to continue to do that with you next year. And if you look at that card, then I would also point out so that there's an artwork on there. And if you look closely, that beetle doesn't have any insect material in it. It's a composite image with plant material. and an artist that we often engage with, who takes her inspiration from the natural environment around us, so something that we all get to enjoy. And Eva's just taken us down a road of really sort of digging into the detail of the biology that we also enjoy. So we celebrate that in this card. And for those in the Fabi community would know that it also kicks off a project where we're celebrating art and biodiversity. And I think I have, there's a YouTube link that we can share afterwards, where there's a series of videos now that capture that and does that celebration. So something to enjoy and share from our world that we enjoy every day with for this season and with your friends and family. So I wish you all the best. Thank you again, Purvis and Tuan, for organizing this, for setting up the technical things in the background, Dave, for hosting today, and Vainant as well. And then all of you, thanks for joining. Eva, thanks a lot again, and I wish you all the best.
Also, thank you very much from my side.
Eva, thank you.
Thank you very much, Eva. Wonderful.
All the best, everybody.
Thank you, everyone.
Keep safe.
Yeah, stay safe, everybody.
Cheers.
Bye.
Thanks, Dave.
Bye-bye.
Thanks, Vainant.
Thanks for joining us, everybody.